Every year, the average adult ingests roughly 250 grams of microplastics. That's enough synthetic material to cover a dinner plate, consumed entirely without notice. Escaping this exposure is practically impossible. Particulates have been identified bypassing clinical filtration entirely, entering the bloodstream directly through intravenous saline drips. The clinical pathology is highly concerning. Recent tissue analyses reveal that concentrations of polyethylene and polystyrene peak at nearly 5,000 micrograms per gram, directly within human brain tissue. This presents a biological paradox. We have to determine how chemically inert synthetic polymers interact with the brain's delicate environment, and exactly what happens when they finally do. Nanoscale plastics do not behave as passive physiological debris. They act as active neurotoxicants capable of breaching our primary biological defenses.
To understand the mechanics of this damage, we need to map the 5 specific cellular pathways through which plastics infiltrate and destroy neural networks. Investigating modern neurobehavioral health requires mapping how environmental debris interacts with the cellular baseline. This 3D cross-section illustrates the blood-brain barrier, the central nervous system's primary endothelial fortress. Normally, tight junctions block foreign particles, but submicron plastics downregulate the proteins holding this barrier together. Suppressing occludin and ZO1 weakens junctions, allowing particles to slip into brain tissue. The second infiltration method relies on biological camouflage, utilizing a mechanism known as the biomolecular corona. When nanoplastics enter the bloodstream, they rapidly absorb surrounding lipids and proteins. This coating effectively disguises their synthetic core from the immune system. As shown in this diagram, a plastic particle acquires a cholesterol-rich protein shell. This specific coating tricks the BBB membrane receptors, initiating clathrin-mediated endocytosis. Because of this receptor deception, the nanoplastic is actively transported directly into astrocytic and neuronal tissues. The brain's ultimate defense system is actively deceived into importing its own destruction. Direct infiltration of the brain is only one vector. The second front occurs through oral ingestion and gastrointestinal accumulation. Microplastics alter the gut's microbial ecology. Specifically, they decrease the firmicutes to bacteroidetes ratio, destabilizing the local microbiome. This dysbiosis compromises the intestinal barrier and generates a surplus of inflammatory metabolites, including lipopolysaccharides. These highly inflammatory cytokines bypass local containment and transmit directly to the central nervous system via the vagus nerve. Therefore, plastic particles that never physically reach the brain can still trigger severe neuroinflammation by weaponizing the gut-brain axis. Once these synthetic plastics breach the neuron, the damage shifts intracellularly, moving past the outer defenses to directly target the cell's vulnerable energy centers. Internalized polystyrene nanoplastics exhibit a distinct preference for localizing directly within the mitochondria. This schematic of the mitochondrial electron transport chain visualizes exactly what happens next. The nanoplastic physically obstructs the sequence at complex 1. This blockage forces an immediate dual consequence: a catastrophic depletion of cellular ATP, followed by a massive cellular spike in reactive oxygen species. Confronted with a sudden energy deficit, the cell triggers a panic response by activating the AMPK-ULK1 signaling pathway. This pathway drives excessive mitophagy. To compensate for the blockage, the neuron is forced to cannibalize its own energy infrastructure. Nanoplastics break neuronal energy homeostasis entirely. They suffocate the cell while simultaneously burning it with oxidative stress. The physical presence of these particles also connects directly to the mechanics of protein aggregation diseases. In Parkinson's disease, the primary pathological hallmark is the misfolding of a specific protein called alpha-synuclein. This molecular visualization demonstrates the interaction. The nanoplastic's hydrophobic surface acts as a physical scaffold, chemically attracting healthy alpha-synuclein proteins, causing them to unfold and bond tightly. This scaffolding effect drastically accelerates the aggregation of these proteins into toxic, Lewy body-like fibrils. To compound the failure, plastics impair lysosomal function, stripping the neuron of its natural ability to clear these toxic clumps away. Because of this specific mechanical scaffolding effect, Patient-derived cell models exposed to nanoplastics exhibit up to a 50% acceleration in toxic protein clumping. Zooming out to the broader neural network, we now observe the immediate aggressive reaction of the brain's resident immune cells, known as the microglia. The persistent presence of synthetic debris forces these microglia into a chronic aggressive inflammatory state known as M1 polarization. In this state, they continuously release cytokines like TNF-alpha. This leads to the pathological pruning of healthy dendritic spines in the prefrontal cortex, directly correlating with cognitive impairment. The most alarming consequence of this toxicity extends beyond the exposed individual. Nanoplastics readily cross the placental barrier. In maternal exposure models, they trigger p53-mediated ferroptosis, an iron-dependent cell death, directly in the developing fetal thalamus and hippocampus. Plastic neurotoxicity is not confined to a single lifespan. It alters the neurodevelopmental baseline of the next generation. This clinical dashboard synthesizes the threat architecture. Plastics induce blood-brain barrier leakage, mitochondrial failure, protein scaffolding, microglial activation, and pathological synaptic pruning. These cellular insults map directly to macro regions of the brain. Damage in the substantia nigra aligns with Parkinson's disease, while destruction in the prefrontal cortex correlates with ADHD and cognitive decline. Laboratory models have repeatedly demonstrated these specific cellular damage pathways. However, establishing direct human causation requires definitive proof. The field must urgently transition these molecular findings into large-scale longitudinal human cohort studies to quantify the exact risk. The primary obstacle for these studies is that a pristine control group no longer exists. Contamination has reached the high-altitude lakes of the Tibetan Plateau. Geographical isolation offers no biological protection. Addressing the exponential global rise in neurobehavioral disorders requires an aggressive, exact accounting of our environmental plastic footprint. The global plastic pollution crisis has evolved into a systemic infiltration of the human nervous system.